The recording is started now. Okay, welcome everyone. Today we'll be having our last PaTaRI of the year, and we'll be talking about histological characterization techniques. I am honored to have my colleagues Mark Eastwood, Adam Shepard, and Mohammed Daoud with me as presenters for this PaTaRI. So before moving on, thank you, Daoud. I really love this quote, by the way, from the X-Files. I don't know whether most of the people in the room have seen it, but it's a TV series about unexplained phenomena, essentially. That's where the X comes from. And in one of the series, there's this quote that what dark work goes on in their inside their impossible machines, cloaked from us by invisible forces. If they know our secrets, why can't we know theirs? This was originally spoken about machines built by aliens, but I also feel that this is very true about cancer cells. What goes on under their impossible machines, and how can we know their secrets? That is what we mean by histological characterization, both at the cell and at the tissue level. Just a heads up, if everyone can keep their speakers muted, we will have no feedback. Thank you. Just moving on. So that's what we mean. We want to go beyond what something looks like in an image to what the tissue or the cell is actually doing within a cancer or within any other disease. So we'll be talking about different histological characterization methods. We'll start from very basic H&E image analysis and then go into very detailed spatial transcriptomics and single cell transcriptomics. How do those methods work? What can they give us? And in between, we would have histological characterization techniques like MRI and also Raman Spectroscopy and multispectral imaging. Now, some of you may have heard about radiology, but those images are very low resolution, but we are going to be educated that you can see cells using CT and MRI. How about what are those technologies? So Adam and Mark are going to talk about that. So that's the brief overview of what we'll be talking about. The goal of this exercise is, first of all, to learn what different characterization techniques are out there and whether we can combine them in interesting ways. So can we, for example, there has been a lot of work going on trying to predict sequence-determined biomarkers from pathology images. Can we do something more in those or similar directions? Okay, so that's the goal. Any questions on this? Okay, before we move on. Now, this is the definition of pathology according to the RC path. And it is, pathology is a study of disease. It bridges the gap between science and medicine. So we want to understand, we just don't want to cure, but we also want to understand how different disease mechanisms work. So here is an abstract framework that I've been able to put together, just some random thoughts, that what we want to capture is these latent disease mechanisms. We want to understand these latent disease mechanisms and latent genetic factors that are in the person. Why do certain people respond to a drug and others don't? That is latent drug propensity. What sort of environmental factors are involved in disease? What are their extraneous variables? What are their epigenetic variables? And nothing makes sense in biology without the lens of evolution. So what are the evolutionary processes involved in different types of diseases? And of course, there are always errors and noise. But the common thing about most of these is that they are unobserved. We can't directly see them, can't visualize them. So we want mechanisms that allow us to infer something interesting about these things by observing some things that we can see. For example, symptoms. We can have clinical observations. We can also see drug effects. So you give a drug to a person, it's essentially an experiment. If the drug works, the experiment works. But if it fails, why did it fail? So that can tell you about what mechanisms are under the hood, what lies under these invisible machines. What is the survival that can also be observable? We can also have radiomic, genomic, transcriptomic, proteomic, or metabolomic information that we can use to infer something about these unobserved phenomena. If we go a step higher, laser pointer is last person. Okay, how do we switch to laser pointer here? It is. Thank you. I'm trying my best. Okay, so that's where it is. So I thank you for pointing that out. I hope you can see it better now. So if something is observable, doesn't mean it's easy to observe. So it may involve cost, for example. So we want to get this information, but and we can observe most of these things, but we may not be able to observe most of these things. Typically, what we do observe is for example, a part of the tissue, an image of the tissue, or a radiograph, or something like that. We can have data from multiple sources. We can have symptoms together with family history. We can have clinical reporting parameters. So those are things that are typically observed. And our goal is to use this to learn representations and then predict, develop diagnostic, predictive, and prognostic models. That's what we typically do, right? Using information that is typically observed. But at a deeper level, our goal is to understand these disease mechanisms. So with that goal in mind, what sort of technologies can help us in reaching that goal? There's also the question about causality. Is what are we observing? Is that really what's happening as well? So, for example, if we see a mutation or if we predict a mutation, that doesn't cause the mutation, right? Is the mutation really there? That is the sort of questions we want to get at. So, moving on. So this is a very abstract level view of how computational pathology problems are. We have unobserved to observable to observed, and then we use the observed information to make models and try to infer latent disease mechanisms. Okay, this is just for completeness because some of the stuff that we are going to talk about actually requires a good understanding of how our bodies work. So I'm just going to give a quick 1-minute summary of how this is. So imagine this is an organism. We'll be starting over here. An organism can be broken down into different systems from the lens of understanding. Okay, each system has different organs, and organs are composed of tissue. Each tissue is composed of cells. Cells have nuclei, and nuclei have DNA, which are composed of atoms. All of us know that, right? Nothing new. The goal is essentially that we want to understand how this works. If you look at the DNA within the nucleus, that is organized in chromosomes, not always, but you can think of that as being organized in chromosomes. And chromosomes are a bunch of protein and DNA wrapped around each other. The protein is called chromatin, and then we have this DNA. Now, this DNA, what it does is it's a biological molecule. Yeah. All of our cells, about 100 trillion of them, each one has DNA except for red blood cells. And this is what dictates how our cells respond to external stimuli, how our cells function, right? So what happens is, before we move on how this code of life is translated, let's just take a quick look at what this code is made up of. So we all know that it's made up of these 4 bases, so adenine, guanine, cytosine, and thymine. The important bit is you see a lot of carbon atoms here, which are bound to hydrogens. This is going to become important when we talk about some characterization techniques. Simon, you got a question? No? Okay. So keep that in mind. Now what happens is, once we have this DNA, there comes, if for example, you have a gene within the DNA which is going to result in a protein, what happens is this big machine called an RNA polymerase comes and it binds to a part of the DNA. And then what it does is essentially creates a photocopy of your DNA in RNA alphabet. So this process is called transcription. So you get an RNA copy of a certain part of the DNA, which is then translated into a protein, or protein is created from that particular piece of RNA using another machine, which is called a ribosome. So once you get a protein, those proteins are the things that actually do a lot of things in your body. So shown over here is a protein. Most of us may have heard the name HER2. So this is a part of the HER2 protein. This is what it looks like. And what it does is, I'm going to talk about that in the next slide. Any questions on this? This is pretty basic, right, everyone? I just wanted to do it for the sake of completeness. Okay. What happens is this HER2 protein is expressed on the surface of cells. So this is the cell membrane, and then you can see this protein being expressed on the surface of a cell. If this protein is activated, I'm going to talk about how it's activated. What happens is that it triggers what is called a pathway. So what happens is that it triggers a chain of events by binding to another protein, which it triggers another protein, which triggers another protein, which eventually results in something called cell cycle progression or mitosis and proliferation. That's what the effect is. And if this becomes out of control, it can become out of control by having too much of HER2s being expressed on the surface of the protein, then we will have a lot of this cell cycle progression going on if our HER2 is activated, right? So you can understand the role of, this is just a single protein. There are about, no one actually knows what the actual number of proteins in the human body is. It may be about 100,000 or more, okay? So this activation over here, if we can stop it by some means, wouldn't that be cool? And that is what this drug, Herceptin, does, which is given to patients whose cancers express a lot of HER2. So to limit this proliferation, what we do is we want to do something to this HER2. This is HER2, what it looks like outside of the cell. So this is the cell membrane, and this is the part of the receptor that is outside of this cell. If it is left like this, it can receive signals, which are essentially other proteins binding to it, and then it can trigger this chain reaction resulting in cell proliferation. Dan, you had a question or something?
Yeah, I was just, is that the structure of the particle, or is that the entire cell?
This bit?
No, the pink.
No, this is a part of the HER2 protein. So this is about 110 nanometers across. This bit. So think of a ball, and this is a small protrusion outside of that ball. So this is the cell membrane. So this is a single protein. Okay. So cells are in the microns range. This is 1,000 times smaller than that. Okay. Just to put things in perspective. Okay, so if some protein comes and binds to it, then this is going to trigger this mechanism leading to uncontrolled, possibly cell growth and proliferation. How do we limit that? We design another protein called Herceptin, which binds to it and essentially shuts it up. So now it cannot receive signals from other proteins that it was supposed to receive signals from, and that essentially cuts the everything, right? Now there are two things to remember in this figure. Where is this protein being expressed? It's being expressed in cell membrane, not in the nucleus. And that fact is going to become important down the road. Okay? The other thing is stopping the action of a single protein may have side effects as well. So if you've stopped it from doing this, doesn't mean it won't do something else. Just like your code has its own way of finding bugs, this is what happens in real life biology as well. In certain cases, if there are a very large number of HER2 being expressed on the surface of a protein, it can deform the cell membrane, which we may be able to see in histology images as well. But those deformations are going to be very small, so this is a paper that talks about that. Any questions so far?
Just to make sure, we have HER2, all of us in our body, and we need it.
Yeah.
Certification.
Yeah.
It is the excess of HER2.
Yeah. So due to an evolutionary event or due to a mutation, for example, this HER2 can be, there can be more copies of it being expressed. So for example, if you have, where did the gene come from, right? The DNA, right? If you have more DNA in your cell, then that is also going to increase the number of HER2 proteins because there is more DNA, then there's going to be more protein, right? That is what aneuploidy or whole genome duplication means. Okay, cool.
There's also a question from David, sorry.
Yeah.
He's asked what are INPY and INPZ?
Oh, those are just PDB structure codes that I use. So 1N8Y is IN8Y is the PDB ID of this structure. So if you want to look at that atomic structure of this protein, just Google this number, and you'll get this protein. Yeah.
How specific is the binding site? Can there be lots of different molecules or proteins that can interact, or is it specific to only?
That's a very good question. There are these proteins called hub proteins. They are pretty promiscuous. So they can bind about hundreds of other proteins. This is just a simplified version of pathway. So this is called, so for example, you have heard about VEGF or P53, right? So these are the effects, these square boxes that you see. This is what happens when there is a change. For example, if this is RAS gets more expressed, what happens eventually is you get more proliferation, but this is a cascade of events. Now, biologists have been putting these maps together, these tangled webs, for ages. There are a number of Nobel Prizes for discoveries included in this chart that you see on your screen. Okay, I don't want to like you to understand, but the main point of the takeaway point is proteins can interact with each other. They can lead to these pathways resulting in things. But how do we find what's actually going on? We can use a variety of techniques. We can use gross examination. Just look at the tissue. Can we see increased proliferation? If we do, is it caused by a certain gene? If we know that, then we can perhaps design a drug to stop that, right? That's the goal. We can also use microscopy. We can have H&E, IHC, multiplex imaging. We can have different types of sequencing technologies. There's label-free imaging. But essentially, all of these technologies that we are going to talk about in the rest of the PaTaRI are a compromise between cost, throughput, and detail that they can give us. So technology can give us very fine pictures of what's going on within each cell, but they may be too expensive for everyday clinical use. So there's a compromise between these three things. Okay, so you can have a solid tumor, you can take a sample out of it, you can put it under a microscope, you can do multiplexed imaging, you can also do sequencing if you have a liquid tissue or a liquid tumor. You can do liquid biopsies with it, and there can be other ways of analyzing it. So we're going to talk about that. This slide is just going to put it so that when we talk about these things, you know what these means. If you can find out all the genes in your body, that is called genomics, right? And that tells us what are you made of, what are you, and what can you do. So if you know what the DNA is, this tells us what can these cells do. Now, remember, DNA gets translated into RNA through a process called transcription. And if we know what transcripts or what RNA is present in the certain in a certain cell and in the body or in the tissue, then that tells us what is the cell about to do, because it's going to make proteins to do that. Yeah.
We heard very much about genetic changes in our whatever gene and so on. So that is related to all 5 of these, or it's just studied in genomics?
Very good question. I'll answer it at the end, of this slide. Ignore the second bit, the epigenomics one. That's related to Mohsin's question. I'm going to come back to it. The third bit, transcriptomics. So if we can calculate what RNA is being made, we can tell what are you about to do. You means tissue or cell or whatever you're analyzing. If we can find out what are the proteins being expressed in the cell, that is called proteomics. If you can get an idea of what are the proteins currently being expressed in the cell, this tells us what are you doing. Okay? And then metabolomics is the field in which you analyze the output of a biological process. For example, if you find a certain increase of a protein in the urine of a person, that falls under metabolomics. And that can tell us, just like your smoke coming out of a factory's chimney can tell us what does the factory do. Like our oil rig is going to have a big flame or something like that, right? That's just a loose example. But that can tell us what have you done. Okay, so that's where. Coming back to Mohsin's question, not all changes in the genes are translated into proteins. So that's one thing. Not everything in the gene, so you're not solely dictated by your genes. There are other environmental factors, for example, that may also influence how your DNA change. They can actually induce localized DNA changes or epigenetic changes that can change how different genes are expressed. So those are called epigenetic changes. So this is what is your environment telling you to do. Does that answer your question?
Yeah, and there are genetic changes as well. That was also part. Like cancer is defined as if there is some gene change inside the cell, then it can develop cancer.
Yeah.
So so we can study both, like in genomics, some changes which are based on purely our genes, and some changes which are based on the.
Yeah, exactly. So for example, remember those DNA was wrapped around in chromosomes. If that loosens up for any reason, the DNA is exactly the same, but now it's loose, right? And that allows this transcription factors to bind and express more, right? If it becomes loose for any reason, due to an environmental change, for example, it's not a genetic change, but it can cause cancer. That changes chromatin accessibility, which is also another technique for tissue characterization. And then we have radiomics and pathomics that most of us deal with every day, and that is what do you look like actually, right? So grossing is essentially, so this is just omics, all of these. So we'll talk a bit more about these. Grossing is the technique in which we just look at the specimen and try to infer what is it doing. You take the body, take the tumor out of a person and look at it just. This is done for two things. One is produce a grossing report. What does it look like? If you just Google or preferably Twitter search grossing, you will find images. I didn't put any gory images here, okay? But the other thing it can be used for is to select a sample that we would actually put on the slide. So this is very important. And currently there is no machine learning method that allows us to do that. There's an open opportunity, guys. Okay. If we have a patient and the patient is undergoing surgery, for example, for tumor removal, we want to make sure that all of the tumor has or has not been removed. We want to verify that. There are two ways we can do that. We can either take a sample of the patient and we can freeze the section, which is called a frozen section, or we can do the whole FFPE slide preparation, or fix the slide in formalin and paraffin. There are pros and cons of both of them. This produces a sample for that you can analyze within about 15 minutes. This takes about 36 hours. So if the patient is on the operating theater, before closing up, the surgeon may want the pathologist to tell them whether they should close them up if everything has been removed. So this is what's being used there. Because this is frozen, but it's just tissue that is frozen, dehydrated and frozen. So this still undergoes very slow biological degradation, and if you scan it under a microscope, you will not be able to identify what different types of cells are. But FFPE gives you much higher quality. Okay, and there is a recent GAN-based method talked about in, I think it was in Fayaz's talk, that you can get FFPEs from frozen sections. So that's pretty cool. But yeah. Very basic, H&E imaging, how does it work? So you've got this compound, this what hemotoxin looks like. Your DNA is negatively charged, this is positively charged. So what happens is if you put this on top of a cell, your DNA is going to be bound, physically bound by these compounds, and when you look at them, then it's going to appear pink or purple or whatever color this is. Right? Similarly, you can have another compound called eosin, which gives a different color, but it binds to things not in the nucleus, primarily outside the nucleus, because this is negatively charged, so it binds to positively charged stuff outside. Okay? So once you have a hemotoxin bound to something, then if you shine a light through it, it would absorb this 600 nanometer wavelength, and the thing would appear anything from violet to blue. That's what this would look like. And if you observe this eosin bound thing, anything that has eosin in it, is going to appear pinkish because it's the cross between red and violet. Okay, that's what we typically do. Then we can also do immunohistochemistry. So this can tell us where is the DNA. If you have more DNA, the thing is going to appear brighter, right, or more purplish or something. You all know that, right? So similarly, this can tell us to some extent how much DNA is being expressed, but it doesn't do a very good job for binding to different proteins. So it has its limitations that I think Adam or Mark is going to talk about. Then we have immunohistochemistry. Do you have seen those brown slides? Where does the brown come from, essentially, right? So it comes from is, so imagine the same diagram that I was showing earlier. Let's say if you got HER2 being expressed on the surface of a protein, then you got this is part of the HER2. What you do is you design a protein which is bound to another protein, which is bound to another protein, which is HRPO, which is shown over here. But this HRPO protein does something very interesting. Whenever this protein will bind to this protein, so this green protein will bind to this red protein, then that means this would also be bound indirectly to that protein, right? So wherever there would be HER2, this would also be bound physically there. And what this HRPO does is that once you put a dye called DAB on it, it catalyzes this DAB into producing a brown stain. That's what you see in your images. So essentially what this tells us is wherever there is brown, my protein of interest is present. Okay? That's how this works, but there's a lot of protein cascading going on over there. Okay, so if you ask the question, is the cell expressing HER2? You can look at it under HE, it won't show anything because it's a membrane-bound protein. What don't we see when we are looking at HE? No cytoplasm, no the membranes, no membranes, right? Because the protein is expressing there, you'll not be able to see any HER2 binding there. But if you do IHC, you'll see membranes where you have HER2 bound. So this is where we can characterize what proteins is the cell expressing, right? So this is an image for a case that is expressing high HER2 versus low HER2.
That makes HER2 more challenging for
Yes.
to be predicted.
Exactly. Yeah. Question.
I've been wondering why don't we just find HER2 directly on a HNA also?
Because HER2 is expressed in the membranes of cells, and membranes are lipids, which are essentially uncharged. So neither eosin nor hematoxylin is going to bind there. So you'll not be able to see much of the membrane within these images. Okay. And you can also use other fluorescence techniques, but I'm going to stop here. There's a question on the last slide, and I'd like to end with that. So what do pathologists do when DAB staining works as expected? It's a trick question.
Sorry, what?
What do pathologists do when DAB staining works as expected? They dab.
Yeah.
Right, so I'm going to just stop there.
So I have a question on this stuff.
Yeah,
So
good.
what they change for different markers
Yeah.
besides this thing? You
Yeah.
just change the primary antibody?
Yeah, this one. Yes, this primary antibody is changed.
The rest is the same?
Yeah, as long as the other part of the protein binds to your secondary, labeled secondary antibody, you'll find. Okay? Who's next?
The antibodies bind to antigens.
Yeah, that is antigen. The protein it is binding to, that is the antigen. That is called antigen. Yeah.
That's a good question.
Sure.
But when would you choose IHC rather than fluorescence?
That's a very good question. I don't know.
Maybe it's when you have multiplex markers.
Fluorescence is not as reliable as IHC.
It is more,
IHC is more reliable.
it's more expensive as well, isn't it?
And fluorescence is expensive. Well, with IHC you can just do it brightly and that's it. Usually, as I understand, the exploration part is more, experimentation is more fluorescence.
And IHC is kind of a surrogate method to detect those DNA changes. So there's always a question how much expressive it could be, the changes that are in. For example, different dyes would highlight different proteins.
Yeah, it's essentially a protein-based marker. It's not a DNA-based marker.
Yeah.
But there are techniques that can tell you about overexpression of certain RNAs using IHC as well.
Similarly, we have been talking about HER2 more here.
Yeah.
So there would be some method that is based on PCR to detect HER2. And it could be definitely, like in MSI, MSS, I know that it's not concordant well with IHC markers. So,
Okay.
like they could 80 to 90% match, but there is difference, disconcordance on some cases.
Okay.
Okay, so as Fayyaz mentioned, the most commonly used methods are H&E and IHC, but these do have some limitations. One of them is that they require quite a long prep time, especially in the FFPE H&Es. That's obviously not so great in an intraoperative setting where speed is of the essence. You can do frozen sections there, but even that still takes sometimes longer than we would like. Those methods are generally only suitable for very thin tissue slices. And taking these RGB images gets information from only quite a limited region of the EM spectrum at quite a low spectral resolution. You just basically have these 3 bins. So there's potentially much more information available there that we could use. And also this staining and associated processing that goes into it can damage the sample as well. So things like label-free imaging, which doesn't involve staining, can kind of help preserve your samples. And over the next slides, we're going to go through a few methods and techniques for yeah.
So I'm wondering about, so most of the time when we try to measure something, that means we want to try to see something, right?
Yeah.
So what would happen if we want to explore the region outside the EM, outside of visible spectrum? I mean, if we can't see it, then does it carry any meaning to us?
Well, potentially it can be, the information there can be used to inform a model which then you visualize with a heat map or something. But there's also options to take your information from more spectral regions and essentially transfer it into an RGB visualization via a variety of methods. But it can also just be, you can be looking within the visual spectrum, just at a finer resolution than just these 3 RGB. Maybe split it up into, I don't know, 10 different bands over the visible spectrum and get more fine-grained information that way. So yeah. So the first thing I'm going to talk about is multi and hyperspectral imaging, and that is basically just instead of imaging with just these 3 RGB channels, you're going to try and image at a higher spectral resolution, potentially, if you want, outside of the visible range, or potentially, as I said, just more bands within the visual range. So there's a subtle distinction between these two. They both involve using more bands of the EM spectrum, but multispectral is generally used to refer to the use of a few extra discrete bands. So there's an example down here. Maybe you've got your RGB in the visible and you're adding a couple of bands in the near infrared and infrared. Whereas hyperspectral is more used when you kind of take the limit of lots of bands very close together, lots of small, very narrow bands very close together. You have something like this where you kind of have, you're basically recording an almost continuous version of the spectrum that you're getting out from that particular sample. So what can we do with it? Anything you can do with RBG, you can potentially do better by multispectral or hyperspectral imaging, just because you have more information there available that you can potentially use if you can figure out how to use it. So some examples of how it has been used. So the first example is multispectral imaging for highly multiplex biomarkers. So essentially what's happening here is they've essentially chosen particular bands of illumination to match with the spectra of the various dyes that they're wanting to image all at once in a multiplex manner on a sample. And you can do this with RGB, but by choosing these specific illumination bands and using more than just those 3 RGB channels, you can actually do a much better job of distinguishing these dyes in the same image and therefore get a cleaner multiplexed image out.
Mark, there's a question by David
Yeah.
saying wavelength affects focal length. Is focusing different colors a problem when making images?
Not that I've found. I mean, I haven't looked too much into the specific technical details of the microscopy setups for some of these, so whether you have to add any extra focusing in there or not, I'm not sure.
So I guess the change in focal length is much less than.
I mean, yeah, when you're talking about
of the imaging system. So
yeah, so when you're looking at a fairly small wavelength range, it's perhaps not relevant, which might be why I've not seen that specifically mentioned anywhere. And then in some of the later things, you're using laser illumination, and there's not that to worry about anyway. So did that answer the question, David? I assume that'll be okay. Sorry? Okay, I'm going to
Mark,
move.
I have one question. For example, in this case, we will get multi-channel image. For example, in case of RGB, we are getting only 3 channel. But in this case, we will get multiple channel, right?
Yeah, I'll explain a bit more about that in the next slide. The next slides. So essentially, what they do is they illuminate the sample with each of these different bands in turn, and take a picture of the transmittance at that kind of wavelength band to get all of these various different images, which they then kind of process all together to give the final multiplexed image, visualizing all of the different dyes that you've put in there. And what you get out is something that looks like this, where you have your various cells stained with your dyes, and due to the fact that you've used this multispectral kind of technique, you separate them quite cleanly and quite nicely. So that's one use of multispectral imaging. I'm going to quickly go to one more, which is learning a virtual staining. They've essentially imaged normal H&E images in 63 different wavelength bands, and then learned a transformation matrix to produce a digital version of Mason's trichrome stain. And on the right here, you can see examples of that, where the left column is the H&E, the middle is the digital staining that their algorithm produces from that, and on the right is an adjacent section stained with chemically stained with the actual stain.
I have a question.
Yeah.
Just a quick question. So make sure, I mean, these slides have been stained only with H&E.
Yes.
And then the digitally stained image has been created digitally.
Yes.
Based on multispectral.
Yeah, based on multispectral imaging of the H&E slides. Yeah. Okay.
So sorry. What's the digital stain that they have generated?
Sorry?
What's the digital stain that they have generated?
It's called Mason's trichrome, is the one they're trying to imitate, and which is the one that you see on this right-hand column. This is the actual histochemical staining using that.
Does it highlight the tumor, or?
I think the idea of that particular stain, I'm not massively knowledgeable on different stains, but I think the idea of that one is to give more detail in the stroma between collagen and other things. But yeah, you'd have to ask a pathologist exactly what that stain is best used for and what it distinguishes. So yeah, that's the section on multispectral and hyperspectral imaging. Do we have any questions from anyone before I move on to a slightly different topic, which is Raman spectroscopy? Okay, I think I'll move on then. So I'm going to talk here about a technique called Raman microscopy. The physical principles I'll go through first of all. So molecules have characteristic vibrational modes which depend on their chemical structure basically. And they can exist in excited energy states corresponding to these modes. Transitions between these states and the ground state can produce signals at particular characteristic wavelengths for a given type of molecule. So when you shine a light at your sample, it will briefly be excited into something that's called a virtual state that it cannot really properly exist in. So it's just a very kind of fleeting state that then mostly transitions back down into the ground state, and you get the kind of more common elastic scattering that, for example, results in the blueness of the sky. That's called Rayleigh scattering. But sometimes it will also fall down instead of to the ground state to the vibrational state. So you'll get a signal at this particular frequency, which is characteristic of this this particular vibrational state. And this sort of elastic scattering is called Raman scattering. Now this spontaneous Raman scattering gives quite a weak signal. It kind of decays randomly in a characteristic short time scale in random directions. It's not particularly great for microscopy. So what you can do to fix that is look at something called stimulated Raman scattering, where you add a second laser beam. So you have now two laser beams at two different frequencies, and the difference between those two beams you tune so that the difference in energy matches the vibrational state that you are wanting to probe. And this second beam essentially couples with this transition from the virtual state to the vibrational state and makes it much more likely to happen. There's a bit of a diagram here illustrating stimulator emission where your incident, this second beam is called a Stokes beam. You have your incident beam here stimulating this transition to the vibrational state and stimulating an emission of a further photon at that frequency. So this technique gives a much stronger signal that you can do microscopy on. I'm going to briefly also talk about a related phenomenon called anti-Stokes Raman scattering, which is essentially when the molecule that's already in its vibrational state sort of interacts with a photon, and you end up with this transition, which is essentially provides a signal shifted by the same amount as the stimulated Raman scattering, but in the opposite direction. One or the other can be used depending on the situation, but they both give roughly similar information and are used in similar ways. And just mentioning that one for completeness. some other non-linear interactions that can be also used to get information about your tissue is something called second harmonic generation, whereby it's a simultaneous interaction of your tissue with two photons, and the emission in this sort of mode actually depends on some of the symmetry properties of your material. So filament-like proteins, and in particular of interest to us is collagen and similar things, will be visible in that mode. So quickly into a typical microscopy setup for this sort of imaging. You have your Stokes beam and your pump beams, two lasers tuned so that the difference between them matches whatever vibrational state you're interested in. It's going sent through the sample, and you detect the stimulated Raman signal. Here you can also detect some other signals in the epi-detection, which basically means photons coming kind of back from your sample. And so, for example, you can detect the second harmonic generation signal there, for instance. So what sort of things can we do with this? Well, these various vibrational modes and frequencies kind of provide different stains in that they react to different molecules and different molecular makeups. So this image on the right is just a bit of an overview of that. Whereas the right hand and bottom ones are various different wavelengths of Raman spectroscopy. And here we have the second harmonic generation and two photon fluorescence, which I'll also talk a bit about later on. But what is of particular interest to us is something called the CH stretch region, which are peaks of the Raman spectrum, which correspond to stretching modes of CH2 and CH3 groups. What's interesting about this is that the mode corresponding to the CH3 groups is stronger in the nucleus, whereas the CH2 group mode shows up more in the cytoplasm. So this gives us the opportunity to kind of generate a contrast between the two, and we can try and create H&E-like images from that, because that's one of the great things about H&E, that it gives you this contrast between the nucleus and the cytoplasm. So this image at the bottom is basically just looking at a zoomed in view of the kind of spectrum of, for example, DNA, proteins and lipids. So yeah, looking at the two signals of these two different modes, the CH2 and the CH3 stretch modes, you can image in these two modes using your Raman microscope, and essentially do some digital staining using, in the case of this paper, they used a lookup table to assign the colours, and essentially get something that looks roughly like H&E. So some examples of that. So on the top here is some images using stimulated Raman histology, and on the bottom are some H&E images.
Just a quick one.
Yeah.
So the main thing is there's no staining in SRH, right?
Yeah, there's no staining here. This is purely probing vibrational modes. So by tuning that difference between the two lasers, first of all, to probe vibrational modes corresponding to the stretch modes of the CH2 groups and then the CH3 groups, you can get a signal which distinguishes between your nucleus and the cytoplasm, as kind of illustrated in these little ones here. And by doing a suitable kind of transformation of those colours, you can get something that looks like H&E without actually doing any sort of physical staining. Yeah.
So if there no staining, then what's in the dark spot in the image? Is that artifact, simply artifact, or something else? On the big image, the black dots. Yeah.
I think that's just some particular colour corresponding to particular signal in the images down here.
So what they did is, as I understand it, they took an image using Raman spectroscopy and they assigned false colors, right?
Yeah.
So the image, this is for false color image, right?
Yeah. So it's a lookup table. So essentially, it's essentially a table matching particular levels that you see in here to particular colours that have been chosen to look like H&E. So no, that will just be some particular value that you see in here, that is mapped to some particular colour that they've chosen.
It's the white spots in the image.
Yeah, the white spot we understand because that's also visible and also with the staining image. But when we talk about the staining, then we usually associate the dark color with the artifact from staining.
Yeah.
Now we see the dark artifact here. But not exactly sure what does that mean in the image.
Maybe it's a clump of genetic material, because CH2, as like where you have more DNA, you would have more of these bonds. So maybe these are just clumps of proteins, maybe, or DNA. I don't know for sure. This is just my guess.
So this does not use any kind of optical objective in the microscope setup that they try to have, like, do they have?
So they have a couple of lasers, and there are lenses involved to focus and things like that. Sure.
Do we know if the resolution of the images, obtained with this setup is like higher than conventional microscopy?
It's a similar sort of resolution. As you can see from these images, they're able to image at a similar sort of detail level as H&E. As you can see from the scale bars here, you can kind of see details down to like 10 micrometers or less.
But the alternate to this is then frozen sections, and we all know how bad those look, right? So.
Yes, frozen section.
So if you were doing it on the operating theater, so your other alternate is frozen sections, right?
Between the LFP and frozen.
Sorry?
How much different the two will look for this technique?
Well, this is not frozen. So this is imaging live there on the, right?
Yeah, what you can see here is the top row. The top row are examples of images using this stimulated Raman histology, so this Raman microscope, and on the bottom are examples of H&E images of similar tissue.
But still from a thin layer of tissue, right? It's not from bulk of tissue.
Yes, it's still fairly similar.
Is there a way to do the tissue directly, to scan the tissue directly without staining?
This is without staining.
It's false colored, right?
But it gets some colors. That shouldn't.
Yeah, this is just a mapping.
Okay.
I mean, the raw kind of things that are coming out are these images on the bottom here, for example.
For example,
the colors.
We could have without any stain, so just to see maybe the morphology and something, and then have two H&E along with, use both as an input for.
Potentially, you could try that, but yeah.
How much do you think would we have any variation in the image? How much will be the variation in the false color? Because in Spain we have a stabilization problem. staining variation problem. But for this, I think perhaps we can apply more standardized.
I mean, there might be some sort of standardization issues in I don't know how bright your illumination is, or potentially things like that. I'm not entirely sure.
Depend on the setup of the light.
Yeah, I think there could still be some form of variation due to your exact microscopy setup, I imagine. But again, the exact probably reduced, I would say, yes, significantly reduced. Yeah.
And is it like high energy beam or?
They're fairly intense lasers, yes.
And
but
how
not.
much do that affect the
It's not.
molecular structure?
Yeah, it's not so high that it will cause any serious damage to your sample. And they're reasonably intense, but not massively energetic photons. Just lots of them, I guess.
There's a couple of questions here.
Okay. What do we have?
Do you want me to read this one?
Sure.
So John has asked if I can imagine it being very good at getting some more insight into intracellular structure and activity. However, surely the additional cost of this is a big barrier to routine clinical use.
Potentially, yes. I'm not exactly sure how much these things cost at the moment. I imagine they'll reduce in price as the technology matures and so on. So maybe even if it's not economically feasible to use them that much now, maybe another 5 or 10 years that might change.
And there could be some other factors too, which are barrier to routine clinical use. Like we have seen one method in the DPC, they have kind of the guy was trying to sell that they have solved this color problem in HME, but nobody so far have accepted to use it. Then maybe the original standard has to be revised. That's maybe barrier.
Do you mean the standardization?
No, the phase contrast microscope is talking about phase contrast.
Yeah, it's some FEI companies.
Oh, yeah, for staining.
Yeah, because speaking about high energy beam, there's a nanotring company which is basically they plastic UV light to perform immunofluorescent multiplexes. And from the image they saw like very high precision in the imaging compared to the usual staining. But then because they plastic UV light in multiple beams, so I'm not sure how much we will spend. The test they didn't really try to answer that question.
On the second question by John, so I think he's talking about the image that we showed earlier.
Yeah, so I've got another question. It might not be super specific to it, in that it could be highlighting DNA in the nucleus and mitochondrial DNA, hence the dark patches outside the main nucleus blob. That's
Listeners ahead were speculating what the other dark blobs were, so that was my guess.
Yeah, that could indeed be right. But again, that's potentially useful information in there as well. So it's not necessarily a bad thing. So yeah, just another example of just a larger scale image using the same technique, with some smaller examples taken from a few particular regions in there. So that's just to show it's capable of larger scale imaging as well. So just going to quickly talk about a paper that was published fairly recently, last year, using this method. So they've used this imaging technique for some near real-time intraoperative tumor diagnostic. So basically, if you have your surgical specimen, the kind of conventional way of dealing with it is kind of going through all of this processing of freezing it, mounting it, staining it, and then looking at this frozen section to try and figure out kind of what your diagnosis is to continue on with your surgery. They've set up and that can take of the order of like 30 minutes or so, which is quite a long time in an intraoperative setting. So what they've done in this paper is made a pipeline whereby this tissue specimen is imaged using this stimulated Raman histology and virtually stained, and then put through a CNN to give a kind of diagnostic indication of what that is. And they've shown that the diagnostic markers, as we kind of saw in some of these examples, can be seen in those images as well as in H&E, and that accuracy using them can be similar to H&E. So
this is 2 minutes, right?
Yeah, this process takes roughly like 2 or 3 minutes. So just to go on a bit more about that. They trained their CNN and Inception ResNet V2 to classify patches of tissue into these 13 different histological categories, kind of subcategories of various different diagnostic classes that are of relevance to their operating procedure. That's to help inform the decision making during the operation, basically. And essentially, they've just got a whole bunch of labelled patches that they've used to train this network. They have these scanners in operation in 4 different institutions in the US. I think the University of Michigan, Florida, I can't remember the others, but a few different places. And they've done a very small scale clinical trial using this, where they have basically a bunch of volunteers that have agreed to take part in this, and their samples during the operative procedure are sent through these 2 different arms: the control arm, which is essentially conventional histological images, and a pathologist providing a diagnostic, and this stimulated Raman imaging with a diagnostic by this CNN that they've developed. And they've got an overall diagnostic accuracy which is roughly comparable between the 2. They've essentially shown non-inferiority of their method. There's a small caveat to this in that they've actually allowed their CNN to choose not to classify a certain number of cases. So they've got a method which essentially looks at the image and says and decides if it's far away from the images it's trained on. So it's basically able to say, okay, this example is outside of my area of expertise, I can't do this. And they excluded 13 particular rare types of tumour using this method. So that's good and bad both together, I guess, because it slightly weakens this result, but it's good that they are able to, using this this AI pipeline, identify what sort of examples their AI will not work on, essentially. And one of the interesting things to come out of this as well was that the mistakes made by the CNN on the stimulated Raman imagery and the pathologist on the conventional H&E were mostly non-overlapping. So there's potentially very complementary methods for improving your overall diagnostic accuracy during operation. So that's that bit. So any questions on that before I move on to the last little section?
We're running low on time.
Okay, sorry. There's been quite some discussions.
That's interesting.
So the last bit is only quite brief. It's just quickly mentioning some other methods that are out there that use a variety of other interactions of various waves with your tissue sample. So there's photoacoustic, where I think that's looking at when your tissue absorbs photons, it heats up slightly, which causes a small expansion, which can cause a kind of acoustic wave that you can detect and attempt to do microscopy based on that. Quantitative phase imaging, which basically provides contrast by looking at the varying refractive index of your tissue, and a few other things as well. So we'll look at a few of those in turn. So the quantitative phase imaging, which is basically looking at variations of refractive index. This one is able to image at multiple depths quite easily without physically sectioning your tissue. So on the right here, you have examples of images of various different depths, which you could potentially, with the right software, stitch together into some sort of 3D imaging system. This paper here is essentially another interoperative use case, whereby they've developed a staining scheme and a microscopy setup, which is again quite quick and easy to set up. So this is a particular rapid dyeing scheme that can be done again in the scale of 3 to 5 minutes, and uses again a virtual staining to transform from the fluorescence imaging from these dyes into a H&E equivalent. So again, here are some example images where on the right you have the sections stained with H&E, so FFPE stained with H&E, and on the left are the images using this fluorescence method and virtual staining to reproduce. And as you can see, the relevant details are there in both, and they do quite a good job of kind of reproducing this H&E stain.
So this gives 3D images in 5 minutes.
Right?
That's the
Pseudo 3D. It can give imaging up to a depth of around 100 micrometers.
Okay.
Beyond that, it's not so great, but it's essentially because the 2-photon excitation allows you to use much longer wavelength light, which penetrates, which has less scattering in the tissue. It allows you to image deeper before the kind of scattering gets too much and doesn't work anymore. So they also have some higher resolution examples of these images. If anyone's interested in looking them, the links are kind of in here. I won't look at them now because we're running short of time. The final one, I think, is some label 3 again, and again an example of digital staining, and this is using a GAN on some autofluorescence images. So they've essentially got some FFP samples and image them in two different frequency ranges. And then they've gone and taken that tissue, histochemically stained them, imaged them, registered those two different channels and the stained images using an elastic translation-based method, and then trained a conditional GAN to basically recreate the stained images from the autofluorescence images, and they get, again, some really, really interesting results. So on the left are the two different autofluorescence channels that they're looking at, and on the right, the far right column is actually histochemically stained tissue. So these two are H&E. This one is Mason's trichrome, and in this column here is a virtually stained output from their GAN, which basically takes this as input and outputs this. Again, you can see
this is again without any staining.
Pretty cool stuff. So this is again without staining. Yeah. So just as conclusions, virtual staining of these unstained samples using this variety of label-free imaging techniques seems to have a lot of potential. It's capable of generating images that look very similar to H&E and do expose the relevant details that you would need for an accurate diagnostic. And an important use case for this sort of stuff is in the interoperative setting, where in particular you can kind of have this time sensitivity. And AI-assisted diagnostic also fits well into this sort of context, as if you can help the pathologist with AI, it helps with decision making in the very short time frame that you actually have. And I'll now be passing over to Adam to talk about a few more methods.
Cool, thanks, Mark. So should I unmute me and then if you...
Yeah.
Okay, I'll start.
Maybe come here.
Yeah. Okay, so where
You're muted. Yeah. Can everyone hear me okay?
better checking out? I think I might not have muted yet. If you check.
There you are. Cool.
Okay.
Cool, so there's loads of different radiological approaches that we can use for generating images. And one of the really cool things, I think, with radiology approaches is that you can do it in vivo. So you don't have to get a specimen, you don't have to get a biopsy, you don't have to cut it up or anything like that. You can generate images like there and then. And as I'm sure everyone's aware, there's loads of different approaches. You've got MRI, you've got CT, you've got planar X-ray, you've got ultrasound. But the two that I'm going to focus on today are X-ray CT and MRI. So just a quick overview really of MRI and CT and some of their advantages and what they're used for is, so X-ray CT, sorry, is based on X-rays, and it's essentially it generates images based on attenuation coefficients in the body. So and you've got an X-ray emitter that spins 360 degrees around the body, and you've got a detector on the other side which picks up the output. So X-rays generate images and they're particularly sensitive to like bone. So in a CT scan, if we see this example CT scan of the brain on the left, you've got particularly high signal in the case of of the skull, but you've got some lower signal in the case of the soft tissue. Whereas MRI is more sensitive to soft tissue, less sensitive to bone. So that's why you can really see quite a nice differentiation between, say, the white and the grey matter in the brain, but not so great signal intensity when it comes to the skull. And here's an example MRI setup over on the right. Now, X-rays are considered to be safe for use in clinic. However, long exposure to X-rays isn't great. On the other hand, MRIs are generally thought to be quite safe as long as you don't wear any metal. So I'm sure everyone's seen lots of images like these before where something's gone wrong in an MRI scanner, left a wheelchair that's not MRI safe, too close to the scanner, things like that. And these things can cost a lot of money when they go wrong. An accident like this can cost about £60,000 to quench the magnet. So it's basically shut down the magnetic field, that is. So a bit of the basis, so we'll start off. So in my talk, I'm going to have a bit of a theme. So we're going to talk about MRI and CT. I'm going to say how they both work, and then a quick approach, how some quick new approaches to advance this to use at high resolution. So MRI, a little bit of a quick physics lesson for the non-physicists, I suppose, is it's based on imaging protons. So protons have a positive charge. These are the nucleus of hydrogen, and protons have an intrinsic quantum mechanical property known as spin. So a way we can think about that in a classical manner is that protons can spin around an axis. And because protons spin around an axis and have a charge, then from electromagnetism, we know that it therefore has a corresponding magnetic field. So you actually have a magnetic field in this direction. So a proton essentially acts as a little bar magnet. So, however, when we place a bar magnet in a magnetic field, then, as I'm sure everyone knows, it tries to align with the magnetic field. So if you've got a compass, then north aligns with north, right? So that's the same thing that happens with protons. When you place it in a magnetic field, the north pole of the central north pole of your proton will try and align with the magnetic field of the MRI scanner. So this is the magnetic field runs through the sensor, through the bore of the scanner. But because these protons are spinning, you actually end up getting a precession rather than just an alignment. So the protons will precess around the actual the applied magnetic field. So that's what this is here. Now, I'm not going to go into too much detail, but essentially, not all protons will align with the magnetic field. Some will align against the magnetic fields. They'll be anti-parallel. So you'll have two states. You'll have a spin up and a spin down state. So a spin up state is low energy. Is low energy, it precesses with the magnetic field. Spin down is high energy, precesses anti-parallel to the magnetic field. And it's similar to what Mark's mentioned a few times in his talk, is that once you've got these different types of energy levels, you can trigger transitions between energy levels via photons, essentially. And that's the basis of MRI signal. It's triggering. You apply a radio frequency pulse, which can cause excitations to higher energy states, and then when you remove the pulse, it relaxes down to a lower energy state, and you emit a photon as a result, and this is the basis of the signal. I've already very briefly said how CT works, but again, it's essentially the image is generated based on attenuation of the beam as it goes through high density signal tissue. So one of the cool things I think I wanted to say really about MRI, and this image here is obviously of the brain, but MRI has multiple different ways to generate images. So you've got structural MRI, which is your standard sort of image that you'll get, but you've also got really cool imaging techniques such as diffusion MRI and functional MRI. So diffusion MRI, you can essentially track the diffusion of water in the brain in this case to generate pseudo sort of white matter pathways or tracts. So this is called a tractogram here. And then on the right here we've got functional MRI, and functional MRI essentially if you ask a patient to think about something, they'll get an increased, or to do something, the part of the brain responsible for those actions will have an increased metabolic activity. And with that, you get a slight increase in the amount of deoxygenated hemoglobin there and then this increases with time. So you get more, and then you get serial blood flow increases, and you can pick this up on an MRI scan. So these.
Sometimes you can have to, for example, if you want to have MRI of the shoulder,
Yeah.
Inject a dye.
Yeah.
Is that what
So this would be structural MRI. So you can inject a contrast, and the contrast will make the scan look slightly different. So that's commonly done it's done for all sorts of things, but in neuroimaging, you do that for imaging tumours, for instance. Yeah. So both MRI and CT, the real problem here is scale. So if we've got an MRI scanner on the left, we've got a histology slide on the right, the sort of resolution you get of an MRI scan is typically about 1 millimeter per voxel, so it's 3D. CT maybe 0.5 millimeter per voxel. If we compare that to a 40x histology slide, we're talking 0.25 microns per pixel. So there's nearly 4 orders of magnitude difference there. There are multiple attempts in both MRI and CT to get high resolution images. So typically human MRI is done at 1.5T. If you look at the brain, it's normally done at 3T, a resolution of about 1 millimeter. A lot of scanners are now, well, there's a lot of work to create higher field strength scanners. So 7T is becoming more commonplace, even working towards 14T. But for instance, this 10.5 Tesla MRI scanner is, by the time you've got the magnet and the ion shielding around the room, it's about the weight of 2 Boeing 747 jets. So it starts becoming quite big and very expensive. So if you want to get a resolution that's even close to that of histology images, then to do this in vivo, we might be waiting quite a long time. But what we can do instead is we can take the specimens and maybe put them through an MRI, put them through a much smaller MRI scanner instead. And if we've got much smaller scanners, we can actually get higher field strengths because it's about the diameter of the bore is the limiting factor to get higher field strengths because you need to fit a human through it in the bigger ones. So similarly in CT, there's multiple efforts to try and get high resolution CT images. So here they've used, so here's an example is synchrotron CT. So synchrotrons are a superior X-ray source to what you'd normally get in X-ray CT. You get much higher intensity X-rays and they can be produced in a shorter time, so you get better signal to noise ratios, but you require a whole synchrotron. So 2 real quick examples here. We've got ultra high field MRI. So ultra high field MRI is anything that's generally classed to be over 7 Tesla MRI, and this is done in rectal cancer. They've taken the specimen, they've scanned it, they've performed structural and diffusion MRI, and this takes quite a long time, nearly 2 to 3 days, to perform this at such high resolution. And they've registered the different scans together. They've performed a few different diffusion scans. I won't go into detail of the different types they've done. I'll just quickly mention one, which is FA. And FA, you can think of, you get an FA value per pixel or a voxel. And FA is a measure of how hindered or how directed the diffusion is. So if you've got complete free and random diffusion, then it's spherical. It's isotropic diffusion. There's no preferred direction. Whereas if you've got hindered direction, there might be a preferred direction of diffusion of water in the area, and therefore you end up getting a sort of ellipse here instead. But an FA of 1 would be complete anisotropic diffusion, and an FA of 0 would be isotropic diffusion. So here's just some sample images of a histology slide. So the one on the right is mass and trichome stain, and the pathologists have basically delineated different regions. So the region described here as A has been it's been described as cancer. And just one thing I wanted to point out here is this is the FA map, and this is color encoded. Sorry, and typically the RGB stands for the direction. So red is diffusion in the X direction. Green is diffusion in the Y. Blue is in and out of the image, so the Z direction. So, but if we, as I say, A has been labelled as cancer. But what's interesting here to see in this FA image from MRI is that actually we've got completely isotropic diffusion. So if the FA is very low, you've got there's no actual direction in this particular image. So I haven't got a pathological explanation, I suppose, for this, but I'll leave that to pathologists, I suppose. Yeah, exactly. Yeah. So, yeah, I'll just quickly show a bit of an example about synchrotron CT as well. So there was a study that used hierarchical phase contrast tomography, so-called HIP-CT. They've used a massive synchrotron in Europe, in France, I believe, and they've used it on the brain, lung, heart, kidney, and spleen, etc. And they've scanned, say, a whole brain in this at 25 microns per voxel, so quite high resolution now. Take about 16 hours, then zoomed in different areas and got it at 1 to 2 microns per voxel, so really high resolutions.
There's a question.
Oh, sorry, Dan. This one?
No. It is after cut out the organs or just for
So-
live human?
So these aren't live, no. These are they're the intact organs, but ex vivo. I think putting a human in synchrotron radiation might have some ethical implications.
That's how you become
But here we've got an image of a lung. So the green volume is the lowest resolution, blue highest resolution, 2 microns. You can see that we're getting some really quite high resolution images here now. Again, some comparisons here now, this time to H&E. So top bottom left image here is a H&E image. Top right of the left set of images is hip CT image, and this is where it's been color normalized effectively up here. And this is when this image here is when the H&E image has been the grayscale version of the H&E image. So for comparison, you can see that actually they're looking very similar, and the resolution you're achieving is quite good. And on the right images here, we've got even higher resolution. And again, you can compare the two different types of images here. But a problem with these approaches, as I've already alluded to, is scalability. So if you need to use a synchrotron, this is a €150 million facility, and each image at 6 microns per voxel was about a terabyte of data. So bear in mind, these are 3D images. So if you want to get even higher resolution, you're talking a lot more memory. I did have some images to show just of a 7T MRI scanner, the resolution you got of a brain, and then the resolution you got of a brain on a hip CT. I won't show you those now in the interest of time, but I'm sure everyone will have access to
What
the.
we can do is, in terms of times out, we can have a separate session on sequencing, right, next time, probably in the next PaTaRI. So, like, everyone's okay to stay for, I think these are a couple of minutes videos, right?
Yeah, they're literally 30 seconds or something. So if this is a 7T MRI scan, and I'll just quickly increase some speed. I need to give you permission for the
It doesn't show cells, right?
Yeah.
They're creating the CD next.
There are technologies which can do 3D. Sure.
Well, this is 3D, but
you write your email in there,
without the slide.
so I can share it with you, so you can use it.
I'm not sure which one it will be.
So yeah, you can see it's quite high resolution there, but if you compare it to, say, which I can't see apparently, the hip CT.
That's downwards.
Oh, this is
anyway.
sorry, this isn't yours. Is that mine?
Yes.
This one's the other one.
Cool. I'll use this one then. It's getting very confusing.
You can share Teams. Share it on Teams. Yeah. And what you can do is, on your laptop, you can show the Teams window.
Yeah, good idea. Okay. Do you want? I'll get it up on here then. I'll share on Teams.
But this is the first time anyone has used an MRI, CT for, or any CT for showing cells, right?
Yeah, exactly. Yeah. And just to give you a background, so that the MRI on the left, that was a 7 Tesla MRI scanner that wasn't that was of a dead person, but it took 100 hours that scan did. So to get such high resolution, it takes a long time.
100 hours?
Yeah.
100 hours, yeah.
What happened to the patient?
I'm not sure, but they weren't alive at the time.
I think it's the expert one.
Yeah, exactly.
Yeah, they know where they get to.
Oh, yeah, maybe.
These kind of conversations.
How do I stop sharing this?
Okay.
And then if I just show you the other example, this is hip CT, and you'll see pretty much instantly. Sorry.
Someone's brain. How do you feel now? Are you looking at colon? Yeah.
So yeah, in comparison, this took about 30 hours to scan, but again, this was ex vivo.
So one of our collaborators is actually involved in this study, so that's how we got into it.
That's much better.
Because it's going through multiple slices, and it's going deeper. Cool.
Where are those cell images? There were some cell level images as well. I think those are on the other slide, right?
Yeah, I think these ones here.
Yeah.
Yeah.
This is part of it. They use it to scan a COVID patient's lung, and that patient's lung.
Yes. Yeah, it's really some really cool. They've got loads of really cool videos for hip CT where they've scanned. They've got the same videos for lung, kidney, spleen, etc. So yeah, I think these approaches got some really cool.
Can you do something like CT? Can you do like MRI and something like?
It's different, I suppose. The limiting factor with MRI is the field strength of the magnet, but with CT, the limiting factor is the intensity of the X-rays you can get. So you get higher intensity X-rays because you're using a synchrotron, so it's more directed X-rays. Whereas it's, as I say, to get higher resolution with an MRI scanner, it needs to be bigger magnets. And these magnets are already getting huge anyway. And like a 3T scanner costs maybe 2.5 million sometimes. So yeah, never mind what a 14 Tesla scanner would cost.
Well, I think we're going to end the session here today. And in the next section, we are going to talk about more sequencing technologies and spatial transcriptomics and other stuff that Doug is going to lead. So thank you, everyone. I hope this was useful. Thank you for your questions. Really appreciate the efforts by Mark, Adam, and Dawood in preparing this. We just want to identify what are our competitors. What are the competing technologies? What are they up to?
What about electron microscopy?
Yeah, that's a good one.
Yeah.
Maybe we can add it to the next one.
I have read one paper they have used infrared imaging, and only this single image they use for tumor detection and saturation.
Great job, everyone. Thank you.